Yes, my name is Pier Andrée Penttilä. I'm the head of the flow cytometry facility here at the LMB. And I'm going to touch upon some of the technical considerations in flow cytometry, and then later on a little bit about fluorescence-based cell sorting, which I think a lot of you will probably end up using, particularly if you're going to be doing any downstream purification of cells for downstream mass spec, as Kat just outlined, or even, for example, purification for downstream cryo-EM, or any kind of future functional kind of screening or high throughput screening. So we'll start first by explaining a little bit about what flow cytometry is. It's a sophisticated cell imaging technology in which a population of cells are hydrodynamically focused. So we work with single cell cells in suspension. And they are focused into a single cell stream, and then they're pulsed with lasers to record fluorescence at different wavelengths. And you can use various fluorescent probes and dyes, so very similar to imaging. And then that allows the analysis of the expression of a variety of different cell markers. And then if you take that one step further, we can actually purify populations of interest using fluorescence-based cell sorting, which applies electrostatic deflection to cells that are placed into drops to purify these populations based on their fluorescent properties. And cell sorting is still the most efficient way to essentially purify your populations of interest with over 99% purification. And I kind of see flow cytometry as a perfect tool or technique for very impatient scientists. So if you want results really fast, then pretty much flow is for you, because one of the things that it does is it gives you results in a very high throughput manner. And so I'm going to try now to essentially cover the main points where fluorescence microscopy differs from flow cytometry, and Nick is going to go afterwards into more detail about this. But essentially, the thing that you need to understand here is that the decision to use flow cytometry, which again measures mean fluorescence intensity, and imaging needs to be driven by your specific biological questions. So always think about that. So the two technologies are not in any kind of direct competition. In fact, they're rather complementary. So they can both be used together to kind of gain a more complete understanding of the cell state, for example, or cellular function. And so in this sense, you, for example, could use flow cytometry to obtain really statistically robust data on a massive or large population, or when purifying a population of interest for any kind of downstream workflows. Whereas microscopy is much more superior, the superior choice when the research question requires any kind of visual or spatial context. So I've kind of tried to summarize the differences here. In terms of spatial context, you can imagine that it's quite obviously high in microscopy, but more or less is lost in flow cytometry because we don't have any of that information. We're just looking at mean fluorescence intensity. But the throughput is very high, so that's an advantage. You're talking about over 10,000 events per second, sometimes even 20,000, depending on the cell or sample that we're sorting. And the information generated is also quite, again, complementary, right? If you're looking for more morphological information, then you're going to go to microscopy. If you just want cell counts and just general protein expression, and you want that information in a rather robust and high throughput manner, then you're going to do flow cytometry. And again, a couple of the experiment examples that I have outlined here, if you want to do really rare event analysis, and again, morphological analysis, any subcellular localization, then you're really going to go to microscopy. But if you need to do any kind of high throughput screening or kind of any bulk phenotyping, and particularly for any cell sorting where you do need to purify populations of interest, the only way to do that is via flow cytometry. All right, so a really basic overview of flow cytometry. It is machines that are comprised of the fluidic and optical and electronic components. Essentially, the fundamental fluidic component, as I mentioned earlier, has the principle of hydrodynamically focusing your suspension of cells. And essentially, we have a core stream, which consists of your sample itself, and then using a hydrodynamic fluid focusing region with sheath fluid, we get the cells kind of focused in a single cell file based on the Bernoulli effect, where eventually they will be pulsed by lasers. What I'm going to do now is just talk a little bit briefly about the differences between conventional and spectral flow cytometry. Some of you may have heard a little bit about this already. So, and it does kind of play into a little bit of microscopy here in terms of principles. So, a lot of the older equipment in flow cytometry was kind of conventional, and that by conventional, I mean that it relies on a series of bandpass filters that are in mirrors that are put in front of detectors to segregate emission of light onto individual detectors. Spectral flow cytometry is kind of the new generation of flow cytometry. And typically that uses a grading system to separate light into a focusing lens before detection. And as the light keeps diverging in space, we typically use a collimating lens to parallelize the light linearly before it reaches the detector. So in the facility, we have these two different types of flow cytometers, both conventional and spectral. But I would argue here that there's definitely more of a movement away from kind of the conventional way of things. And I'll explain and show you a few slides as to why that is the case. So I mentioned earlier a little bit of the differences in optics. But again, spectral captures really the broader. I don't like when people say full spectral, because you can't technically get everything fully spectral. That's not possible. But I would say a broader emission pattern of a molecule. And it can be done, obviously, as I mentioned, by light dispersion techniques. We don't really use prisms anymore. It's mostly grading. And we have a machine called the ID 7000. And that's what applies it applies a grading system. So if you're looking at kind of typical flow fluorescence emission plots, this is an example of, let's say, a green fluorescence emission, which follows kind of a semi-Gaussian distribution. And you can start looking at multiple different spectra of particular proteins. So if we do that in a more conventional way, you can start to see that there's some overlap between, let's say, your green fluorescent protein, maybe something in the yellow range, and then again in the red range. So that's how it typically looks. And what you would imagine is that if you have two fluorophores here and here, that in reality, the way it's actually read is more like this. Okay, so there's a summation because of that spectral, what we call spectral overlap. And then between the red and the rest, you're going to have more, again, a summation or an addition of that spectral spill, as we call it, into the other detectors. And so what conventional does is it would apply a filter in the peak emission range of whatever fluorophore you're looking at. And then to deconvolute those things, it would basically be only looking at that part, that peak emission of the spectrum. So it's essentially not looking at any other parts of the spectral emission of those particular fluorophores. And so when we place that bandpass filter in front of each of those detectors, that's essentially what we're doing. We're discounting any of the other information of that emission profile. And what that translates to in a real terms is this mean fluorescence intensity. So for example, here in the green channel, you have about 85,000 mean fluorescence intensity. And then in the orange channel or yellow channel, you're going to have a little bit of that spill added over from the red, and then a bit of the spill from the green as well. So it would be a little bit higher. And then again, in the red, you're going to get some contribution of the orange and the green into the red. And so what we would do is we want to ensure that each of these contributions of spectral spill are removed from that initial readout. Because if you don't remove those, essentially what you're facing is potentially some false positives, for example, that don't truly exist. They're not really real, but it's just positives based on spectral spill or emission spill into those other detectors. So when we do what we call typical spectral unmixing or compensation, as we call it in a machine, is that we would then remove that mathematically. But then what happens is when you do remove that, you're actually removing a bit of the sensitivity in each of those detectors as well. So you have to take that into consideration when you're doing your panel design. So ideally, in an ideal world, you would avoid kind of any fluorophores that overlap. But realistically, most of you or some of you will actually have some level of overlap. So you'll have to consider that. In terms of the spectral view of things, you still have the same problem, but instead of actually looking at just slices of light in the peak emission profile, what you're doing is you're getting a summation of all of those peaks together across the wavelengths, the visible wavelengths. And so ultimately you get this kind of full spectral view. And using a weighted least squares unmixing method, you are actually fully unmixing all of those various profiles from one another. And again, you're still going to face certain reduction in sensitivity and readouts because that's just the way it is. But the advantage here in spectral is that you have the ability to use two fluorophores that would be very similar in terms of their overlapping emission profiles, but you would never be able to use in a conventional cytometer because they would be actually directly overlapping, and it's not taking into consideration any subtle differences in their spectral profiles. So it really gives you kind of more of a broader ability to use, let's say, two fluorophores, something like GFP, green fluorescent protein, and Alexa fluor 48, that you wouldn't be able to use in a conventional machine, but you would be able to spectrally potentially unmix in a spectral system. So the other thing that I really want to emphasize about spectral that's really an advantage as well over the conventional older systems, there's a few. So the other thing that's really good is that when you're taking your fluorophore and you're binding it to whatever protein, maybe potentially you're looking at a certain antigen and you're using a tandem dye. I think Nick will potentially discuss a little bit about tandem dye, so I don't want to talk too much about that. But if you're using a tandem dye with a donor and a receptor, sometimes the tandem dyes fall apart. And in conventional flow cytometry, you actually wouldn't be able to necessarily see if it's starting to fall apart. In a spectral view, because you're getting the full spectrum of that particular fluorophore, you're able to see if the tandem is falling apart, if it's starting to actually excite and have a cross-beam excitation at different points, and then ultimately extra emission across the range of wavelengths. So it's a way of essentially quantifying any problems with your tandem dyes. Potentially you're fixing your samples, and that also will cause some kind of tandem dye degradation. You may want to assess how much is happening there. So that's one of the advantages of spectral over conventional. A second really big advantage is the readouts of bare autofluorescence. So if you're actually looking at a mixed cell population, for example, peripheral blood mononuclear cells where you have lymphocytes, monocytes, maybe granulocytes, they're all going to have varying levels of base autofluorescence or background. And that's based on their inherent cellular properties. Granulocytes, for example, neutrophils can have a very lobular kind of nucleus. Lymphocytes are just basically all just solid nucleus. So they look very different actually on their scatter profiles, and that actually also translates into very different levels of autofluorescence. And so a really a nice aspect of spectral is that you can run your unstained cell type, and you can actually deconvolute it or look at its spectral signature, because every cell will have an inherent spectral signature, and then deconvolute that from the rest of your other cell types. So this can be a really big advantage for people that are working with kind of mixed cell populations, particularly from primary tissues, but I think it also applies equally to any other kind of cellular types. So again, really great advantages to spectral cytometry. So I'll talk a little bit about some of the measurable parameters, because this is probably the thing that you're most interested in. So I mentioned this already, but a little bit about cell surface and intracellular antigens, protein expression, any phosphoproteins. A lot of people do phosphoflow cytometry. We can look at total DNA or RNA content. You can use fluorescent proteins, apoptosis and cellular viability. And before I go on about total DNA content, when I talk about that, I mean more cell cycle analysis, any kind of cell kinetics, proliferation, or ploidy or aneuploidy. And also we can do chromosome analysis in the facility. We actually did a lot of this for Julian Zale and Jason Chin's lab, where we essentially purified out chromosomes for downstream analysis. And then as well, you can do any kind of like organelle marking as well, for example, mitochondria. All right, so moving forward, I'm gonna kind of start talking a little bit more about cell sorting, how it works. We have different systems available in the facility. The first is under the A plot here, or A figure. This is just showing how a typical analyzer works. So you have your cells, you have a sample inlet, they're taken up through their pressurized system. They go through a quartz flow cell, and then we have the interrogation point where you have the laser, so the cells are pulsed with the laser, and then essentially the sample goes to waste. So in this context, you wouldn't actually be sorting your cells, you would just be analyzing them for any kind of fluorescent readout. And when I say interrogation point here, I should make a point of talking about how we use various different lasers. So it's not just one laser, we can actually use anywhere from 1 to even 9 different lasers. So it really gives you kind of a lot of flexibility in terms of fluorescent readouts that you would like to take. So when we talk about cell sorting, that's a different story, because what we're doing is we're still flowing cells and hydrodynamically focusing them, and we're still pulsing them with lasers. But what we're doing is we're then actually purifying out those cells by placing them in droplets, and then for any kind of downstream analysis. So the way that works, and we have 2 different types of cell sorters in the facility, and essentially there's 2 different types out there right now, is we have both the quartz cuvette-based system, which is kind of similar to the analyzer that I've outlined here, where the laser interrogation point actually takes place essentially above the exit orifice. So what this means is that there is, using a quartz cuvette-based cell sorter, is you get a little bit better resolution. And then what we have in figure C here is an example of what we call a jet and air type of sorter. And in these instances of these types of machines, the cells, once the droplets exit, they're actually, the laser interrogation point is below the nozzle orifice. So what this essentially means, and it takes place within midair, is that we can sort a little bit faster with these systems. So they both fill a niche. I would say that if you wanted a little bit more resolution, then you would go with a quartz cuvette-based system. And if you wanted a bit more speed and high throughput, then you would go with a jet and air system. And above this, these two cell sorters, what we have is we have a piezoelectric transducer that's vibrating at a really rapid rate. We can actually change the frequency of that transducer anywhere from typically about, well, quite low to 10 kilohertz, let's say, all the way up to 90 kilohertz. And that means that we can essentially control the size of the drops. We also have different nozzles. I'll touch upon that in a second. But we can essentially control the frequency. So that's the speed of sorting. So obviously, if you dial up your frequency, you're going to have many more drops produced, which means you can sort at a much faster rate. If you go way down, the opposite, you're going to be sorting a lot slower. Your droplets are going to be bigger. So again, differences for different applications. And then when these droplets are broken off from the mainstream, essentially they're given a charge, either positive or negative. They pass through a series of electrically charged plates, and then they will be sorted with the opposite charge into collection tubes below. So it's highly, highly efficient. It produces very high purities. And the great thing is that we can sort in a variety of different collection devices, tubes. We can do cloning. Single cell cloning is very, very efficient for that. And again, you can get a lot of high throughput, a lot of cells very, very quickly. We routinely have our cell sorters are booked pretty much all day, and we go through in a typical week, we'd probably go through potentially over hundreds of millions of cells in a week, which is pretty amazing when you think about it. I've been doing this for a really long time, and I'm still every day amazed at this technology, which was developed actually quite some time ago and is still going and still really efficient. So I mentioned a little bit about nozzles, and this is really important vis-a-vis your cell types and your particle types that you actually would want to purify up. And the reason why it's important is because you're going to have different sizes. You're going to have potentially, things like bacteria or even archaea that we analyze in Buzz Baum's lab, which are very small, less than a micron or about a micron, anywhere up to, things like lymphocytes or granulocytes or any kind of cell line that's much bigger. We can choose a various nozzle sizes to kind of suit the specific needs of the particle that we're sorting. And an important aspect of this is that we need to consider both the size of the particle, because you can't force a very large particle through a very small nozzle. You're obviously going to cause some shear forces and undesirable effects on viability, but also it's just not going to sort very efficiently. But you also need to weigh and balance the speed with which you're sorting. So we kind of have to balance both of these things. The typical nozzle sizes that we use in the facility are kind of 70, 85, and 100. We do have on the Bigfoot sorters much larger nozzles, 120, 150, and there's a new 200 micron nozzle coming out. So we use much, much lower frequencies, which means the droplets are quite big. And so there's going to be more volume generated in terms of liquid. But it does mean that for very sensitive or very large cells, that we can sort them. It just means that we won't be able to sort them as quickly. So do come and talk to us. Let us know your needs, and we'll have to change the nozzle size and according to what your needs are. All right. I am going to kind of go now into some future developments in cytometry that I think are really, really exciting. In particular, some of the things that we're working on in the facility ourselves. And that I hope will be a good segue for Nick who's going to give his talk on imaging. So some of you may have heard about imaging cytometry. I hope some of you have anyway. But we're even now going into kind of the next generation of imaging cytometry, where we're now merging imaging and cytometry, but with cell sorting, which is a really exciting time in the field. Kind of the big paper that came out in 2022 by Schreevogel in Science was kind of this really groundbreaking thing, because it merged basically really these crucial components of having some kind of image using a fluorescence imaging using radio frequency tagged emission in a traditional cuvette-based sorter that I mentioned earlier. So there's no jet and error kind of imaging sorters, at least not yet, with low latency signal processing and sorting electronics. So this is pretty amazing, because if you think about it, you are imaging a cell that's quickly moving and through a stream at very, very rapid rates, right? So this has been kind of a game changer, because now the field is really moving towards, like I say, trying to merge all of these things together, getting a blur-free image, moving more towards more higher and higher resolutions, okay? So we can't use this system to necessarily sort, 20,000 events per second. This is an image from the paper that was published, and they ended up using this for CRISPR screening. So you can see that, the images are not super high resolution. Okay, so that's one thing. So there is always a trade-off with these things. But it's pretty incredible that you can sort up to, they say 15,000 events per second. It is about that, I think, with this particular pipeline. But where you're able to get this kind of resolution, and at the same time, you're getting an image and you're actually purifying up populations of interest. So now you're able to actually link more morphology. I would say morphology is maybe a strong word here, but at least some kind of, imaging phenotype with a purified population of interest downstream, which is, I think, going to be very crucial for things like CRISPR screening, and even downstream from that, CAR T cell therapy, stuff like this. So I think it's going to be a real game changer. So this one is at a flow speed of about 1.1 meters per second, which is basically the general flow speed of cells in cell sorting. So there are a few, I would say, let's say, limitations to this technique. The first is that the image is not particularly of high resolution. And another disadvantage of this system is that it only has a blue laser for the imaging side, and I think it's 3 detectors. So you can do a couple of different dyes, but it's still limited in terms of laser excitation points, and kind of multiplexing more than one or 2 dyes together. You can't, at least not yet, I think it's coming though very, very soon. So you cannot also use 2 dyes on the imaging component that would overlap, because there's no way of currently spectrally unmixing them. That's coming now very quickly, and that will change very quickly. Okay, so what are some of the applications that we're kind of going to be thinking of in the future in terms of imaging cytometry and imaging cell sorting? The first is kind of merging cell painting, or a kind of morphological profiling in terms of applying different drugs to cells and being able to link that back to morphological changes. So currently that's done with high throughput microscopy. What would be a real game changer is if we could apply this and not only do, get an image, but also be able to kind of downstream sort these different cells for downstream analysis. Obviously, I've mentioned this earlier, but high throughput functional genomic screening. We could do things like quantitative analysis of internalized bacteria and protozoan parasites, anything in terms of evaluating asymmetric cell division, And particularly, I think this would be a really interesting analysis of cell interactions and at the immune synapse. So I think it's a really exciting time. Things are kind of rapidly changing in the field. In the facility, we have Bill Flanagan, who's working between myself and James Manton, who is building a very kind of high resolution OPM cytometry system. So look out in our space. You can come and talk to Bill if you have any kind of projects that you might think, oh, maybe this would be interesting to test on his system. So definitely come in and chat with us. We'd be happy to discuss any of that. So yeah, look out in this space because I think it's going to be a really exciting time, particularly for all of the new students that are coming out. I think you know, look out in 5 to 10 years. I think things will change quite significantly here. So, and all for the better. So, as I mentioned, current limitations. This probably should have gone before, but so these are the current limitations of the imaging cell sorting. Certain applications will require cell fixation. I'm not really a big fan of that, but I suppose in certain instances that can be okay. But ultimately, it does require people to have an in-depth knowledge of both imaging and cytometry modalities. So again, I think this requires people in both fields to be kind of working more and more closely together, particularly on the cytometry side, so that we kind of stay abreast of any of these changes in technology. And as I mentioned, I think the next frontier is really high resolution, multispectral. So that's using more than, you know, one fluorophore, maybe up to 5 or 6, as James and Akash have recently developed, and applying that to imaging cytometry. All right, last slide. This is the flow facility team. So we have Bill, Sammy, Aggie is our degree level apprentice, and James. And we're obviously here to make the most of whatever you need. Okay, so we really relish working with challenging samples. I mentioned that we work with a range of different samples across the LMB, so we really service all 4 divisions. It's not just we're technically embedded in the PNAC division, but really we're quite, you know, broad ranging in terms of helping everybody across all 4 divisions. I always find it odd that cytometry is not in cell bio division, right? It's a bit strange, but anyway, yeah. So we have a depth and breadth of expertise across various technologies and applications. I want to also stress this too, that, and I mentioned it briefly at the beginning, but if your experiments, really ultimately your experiments are part of a larger pipeline, right? So normally people will come to us first to kind of purify up populations of interest for downstream mass spec, whatever it is, proteomics, any kind of imaging, especially cryo-EM. Come and talk to us and consider the power of cross-facility communication. This is really, really key. You know, Kat mentioned it earlier, come and talk to us. We're the same. We're here to help you guys. But in particular, if you're like, oh, I'm thinking about I want to purify up cells of interest to do mass spec, then that's really important that I know that and that Kat knows that, because we work together, we communicate, and then we get your results in a more quick and efficient way, right? Rather than faffing around and not really knowing what you're doing, it's easier for us to know in advance and to talk together and get you the results that you need, okay? And finally, if you are using the technology, I think this is really important, please make sure you credit the facility in the acknowledgements of your papers. I cannot stress this enough. It's really important because we write grants. Very annoying. The facility people have to write grants to get kits, right? One of the few, you know, people here that have to do this every single year. It's a lot of work, but it makes it easier if we can show the papers that have had really high impact using the technology that we currently have to get more, right? So make sure you acknowledge us. And also, I just think it's the correct thing to do, right? We're here to help you. You don't need to be an author on your paper, but to acknowledge the technical expertise of the people that have helped you along the way, I think is the correct thing to do. So thank you for attention, and I'll pass it over to Nick.
Right. Can I image it? So now we're actually looking at the spatial details, but actually, thank you for all the background work from Pier about fluorescence, because that makes my life a lot easier. So yeah, first of all, I just thought I'd introduce the team. So there's a whole bunch of us working on microscopy, light microscopy. It's sort of split into two halves, roughly speaking. Jerome runs the image analysis side, and he's working along with Dina and Simeng. So they're basically dealing with the product of the instrumentation. Me and Sarah and Thomas are working on the microscopes. We'll train you how to use the microscopes. We keep the machines working. So please do come and have a conversation with us. And just as Pier mentioned, we are talking to each other because we need to know where things are going wrong in the analysis so that we can go back to the microscopes and fix it. All right, so can I say really what we're doing is we're more doing imaging across scales, looking at the two-dimensional or three-dimensional relationship between components. So we're going all the way from animal models, bits of mouse, right down to the subcellular organelles in the single cells and down to single molecule studies. Problems we've got for light microscopy, we've got a resolution limit, which is down in the region of the subcellular organelles. Up this end, it's scatter and absorption, just like I can't see through my hand. That stops us doing microscopy in big lumps of tissue. Before I get into all of that, I thought I'd just unpick a bit the distinctions between magnification and resolution, because these two things are quite important. And in particular, it helps you not collect too much detail when you don't need it. So in light microscopy, we talk about this thing called the numerical aperture, which is defined by this angle alpha, this half angle here. And then we talk about this thing called the numerical aperture is the refractive index times the sine of that thing. And that property basically determines the resolving power of the microscope. So Abbe, when he started looking into this problem, the difference between magnification and resolution, came up with this formula here. So the resolution is inversely proportional to aperture. So it means we want this number to be as big as possible because our lambdas are quite large. Our lambdas are 500 nanometers or something like that in the visible spectrum. So this is what sets us apart from EM, for example, where their lambda is tiny. So their resolution is tiny. Their NAs are very, very small, in fact. And the other point is the refractive index can modify that. So if we use an immersion media, 1.5 oil, that number gets bigger. So what's going on? If we have a point source, we put it through an aperture, in this case a lens, but just consider it an aperture, we get a diffraction pattern at the end of it. That is just an inescapable part of wave physics. Now, for most optical instruments, you don't care. A pair of binoculars, that is not the limiting fact. For a microscope, when we're trying to see the finest detail possible, this is the limiting point, and that's what that sort of looks like in 3 dimensions. This we call the point spread function. And this is, in this instance, the lens itself is actually forming the far-field diffraction pattern, and that's what's going on in the microscope. And so the resolving power is very, very strongly linked to the size of that function there. So, we've got 2 point sources. As long as they're far enough apart, we get 2 of these peaks. We can tell them apart. Once they get very close to each other, they merge into one, and you can't really see the difference between them. But the point here is the size of those peaks, which is determined by the NA of the lens, is going to determine when you fail to start distinguishing those objects. So here's some cartoons from real, scaled from real lenses. So actually, having said that, that lens is somewhere up there because its working distance, relatively speaking, is wrong. But you can see what happens as the NA goes up, as this cone angle gets bigger, PSF gets smaller and smaller, and these are relatively drawn about right. The other thing, in order to get high NA, you have to sacrifice working distance. Otherwise, these lenses will become phenomenally wide, and that's just counterproductive in a microscope. So when we go to the highest NA lenses, like 1.4 NA oil immersion lens, the working distances are tiny. You can only work with cover slip samples. You cannot use this lens to look through the bottom of a 1 millimeter plastic cell culture dish. That one will happily do so. That's worthwhile keeping in mind. Okay, so there's a white light, a particularly poor one. Actually, I was surprised how bad that was, but that's what I got. There's no contrast in that at all. There's not much useful information in there. We can do phase. Now we can see the details. This is what typical cell culture microscope might look like. You can definitely identify stuff. There's a nucleus and whatnot, but we don't really have much, any specificity there. What we want to do is really increase the information content of light microscopy by adding contrast to multiple specific molecular species in the sample. That's what we can do using fluorescence labeling. And we've got sort of antibodies against proteins, toxins that stick to particular structures in cells. We've got binding partners, GFP, we've got lipid labels. We can label RNA and DNA and whatnot. So we've got a whole toolbox of things to label specific proteins. And fluorescence is the way we see them, one distinguished from another. So the advantages of fluorescence, we've got good resolution. We are really down at the subcellular level, no problem. High sensitivity, we'll get onto that slightly later. Very high specificity. If you tag a protein with GFP, basically when you see green, you are seeing that protein. And most importantly, this technique is compatible with living things. And that's one of the things that sets it apart from some of the other techniques that are available with this. You can look at live stuff behaving on the microscope. Fluorescent microscope is not that complicated. You have an LED source somewhere back here, goes through the microscope into these sort of filter cubes here. So this consists of 3 components. We've got an excitation filter, so that's the thing that selects the right wavelength of light to excite your fluorescent label at the sample down here. We've got this mirror, sort of a wavelength selective mirror, so it reflects the excitation light down here, but the emission from the sample, which will be shifted into a longer wavelength, so blue becomes green in fluorescence, that goes straight through there, and that goes up to the eyepieces or to a camera or what have you. And as Pier has amply introduced to us this idea of spectroscopic labeling and whatnot, we've got all these different fluorescent dyes, just like having flow, and you can multiplex. And so typically on most microscopes, we've got a sort of blue channel, a green channel, a red channel, far red channel, something like that. You can pretty much expect those. So that's really helpful. So very simple technology allows us to multiplex different things. So we deliberately don't use dye names when we talk about these because there are loads of dyes that are green and they will pretty much all work on the microscope. And there are lots of dyes that are red and so on. So there's not much choice in the far red because this part of the spectrum at the moment is sort of out of reach, but that is probably changing. So this is how we do multiplexing, just by labeling things in different colors and using some sort of spectral separation to get the image. The other thing that's useful to bear in mind is how big are these things. So this is my terrible color. So here's an organic dye, which is sort of 1 nanometer or 2 nanometers across, depending on sort of what is complicated. That's the sort of smallest thing we can put onto a molecule that's fluorescent. Once you get up to GFP, we've got sort of 3 nanometers by 4 or 5 nanometer beta barrel thing. A small protein, streptavidin, I don't know whatever that is, 40 kD, I can't remember. That's a big blob like that. these so-called quantum dots themselves are actually quite large. They're typically sort of 10 nanometers across. And then we get to antibodies. These are massive. That's a good sort of 20 nanometers across that thing. So that can be much, much bigger than your target protein, but nonetheless, these are staggeringly useful, and we can cover them in fluorescence as well. So that's quite handy to make things show up. But where does that take us? This is the same sort of sample, the same sample image with a bunch of different lenses, just so you can see what their resolving power is like. So if you're doing single cell studies, probably you're going to be working with actually more typically for a fixed sample like this one, an oil immersion lens, and you get diffraction limited detail of the bits and pieces of mitochondria. You can see the cytoskeleton. If you are screening a whole brain slice, very, very unlikely you need detail at this sort of level. So don't over collect, I think, is the message here. Try and get it, suitable. This one is a really good happy medium, and this particular lens is still an air lens, so you get lots of resolution, but because it's an air lens with a reasonable working distance, this is very, very conducive to automation. So you can do a lot of hands-off work with a lens like that one. We have those sorts of. Okay. But in the limit, this is a sort of detail you'll get from a 1.4 NA oil immersion lens, something like this. And this has got 4 different fluorescent labels on. These labels happen to sort of illustrate quite a lot of the properties of fluorescent dye molecules that we use. We got this thing called DAPI in the blue, which is a nuclear stain, but that actually is a small molecule that directly binds to its target of interest, and it happens to be fluorescent. So that's a sort of a direct binder. Then we've got 2 different antibodies. We can multiplex antibodies. We've got sort of a green labeled antibody and a red one. So you've got primary, secondary. So here, the limitation sometimes is actually we've got to have these raised in different species. So sometimes you just cannot get enough species diversity to get the labeling you want. But other than that, it's okay. And then cyan, this is actually a toxin from a fungus, and that's what makes it poisonous. And that actually binds directly onto the actin cytoskeleton and causes us lots of problems if we eat them. Just at this point, I really wanted to emphasize, if you're going to do downstream analysis of all that, you really need to think about some sort of housekeeping channels. You can't, you shouldn't really be doing your analysis based on segmentation, for example, on an experimental variable, because if your variable drops to 0, you no longer can segment the thing you want to segment. So housekeeping channels help you find the stuff independently of what you're actually doing to the system. And DAPI is often used for that purpose in fixed samples. And then finally, just a reminder, GFP, the fluorescent proteins are an incredibly valuable and useful tool in microscopy. They're available in lots of different colors. And for me, I think, what the hell did people do before GFP? It's really quite difficult to imagine how we worked without them. But they're there. Okay, I can stop here now. I told you all about microscope. Really, there are a few considerations. This is where we can help you. So there are lots of things here. I've realized I've missed a few bits and pieces off this list, but live samples, fixed samples. Live samples have their own set of constraints that you have to work within, whereas fixed ones, you can do a lot with them. So live one of the primary things is, what about the dimensions of time? How fast is the thing moving? How quickly do I have to image it in order to properly capture that process? Space, I'm imaging a whole mouse brain. Do I really need to see every single bit of the mitochondria? Probably not. So those are important considerations. We can also sort of speed things up on the microscope by doing multiple positions in a sample, doing tiling, all that sort of stuff. So there's lots of spatial dimension consideration. The spectroscopy, so Pier really introduces very nicely on the flow. We have the same constraints. What are the number of channels? Here, what we're all talking about is the molecules of interest. How many can we squeeze into the machine? What about if we're using dyes that act as sensors? So dyes that measure pH, chloride, calcium, you name it. Those have got their own spectral properties. I've talked about fluorescence, but actually there's this whole business of histopathology. You know, we can just do H&E staining and see what we need to see if it's a piece of tissue many times. So there's transmitted light microscopy for that. There's all the genetics you have to do, fluorescent proteins, knocking them out with CRISPR and stuff like that. And all of this, we can apply automation to in terms of the imaging, rather. So if you've got a big box of slides that you want to get through, it's going to take you hours to grind through those one by one. If you automate the machine, you can just load it up and go. There needs to be any drug treatments, any other sample intervention, and finally ethics and biosafety. You have to deal with that. So that should be dealt with by the time you get to the microscope. And then all of this lot sort of leads into how you choose the optimal microscope. So here we go. Here's a wide field. Here's a wide field image of cell in 3D. Great, lots of details. What you probably can see is that there's a sort of haze all around this cell. So it's not so bad on a single cell, but if that was a piece of tissue, that out of focus fluorescent haze completely obscures the details you're trying to see. So we have a great fix for that, which is confocal microscopy. Yeah, that's a confocal microscope. The confocal bit is that box on the side there. So that essentially is just a point scanner. It scans the sample with a fixed focal point. The fluorescence emission goes through a pinhole onto the detector, and it generates sort of images on the screen, just as you see there. What the pinhole does is reject all that out of focus fluorescence, so you end up with a much more contrasty image. So here the haze is gone, and also because of the pinhole, for reasons I won't get into just now, can squeeze a bit more resolution out of the sample. There are other ways of doing sort of this contrast enhancement. So we can do it computationally using deconvolution. So here, these are live bacteria. There's the scale bar, 1 micron, they're tiny. We can deconvolve the image and see small details. So the challenge here is to do this volumetric imaging fast enough that that detail is not smeared out by motion blur. But that's sort of at the limit of what we can do with deconvolution. Right, I'm just going to go into imaging in tissue. So here we've got to deal with ever-increasing scatter and absorption, but scatter is particularly bad. So the goal is to observe processes in physiologically relevant tissues. So this is where, this is some work of the Lancaster lab. So these are brain organoids. So these are recapitulating a lot of the functions of, a lot of the anatomy of the brain, but in a dish. But as you can see, this thing is a sort of milky white. You just cannot see through that thing. That blob there is a sort of outgrowth that recapitulates a lot of the structure of the cerebral cortex of the human brain in this interest. So it's a really fantastic experimental model, and it's genetically tractable and everything else, but it's already grown too big to image. So this thing could be a year old and many millimeters across. So what is really interesting about Madeline's group is that they recognize that this was a limitation. And basically they re-engineered their sample to make it tractable for imaging. And this is quite a really unusual sort of thought process in microscopy. What can I do to make it work on the microscope? So what they're doing are these slice cultures. So you can still just about see these stratified cell layers. And this is called an alico, or air-liquid interface cerebral organoid. They then put that thing into a micro-patterned sample chamber. And the question they're trying to look at is, do the neuronal bundles that they see in these objects exhibit coordinated activity? Because that's a sign that this thing is doing something other than just growing. And the other thing that they do is they put in, they're doing functional imaging. They put in a genetically encoded calcium sensor. And this sensor is just an example of one of the many, many, many different flavors of these things that you can use, called GCAMP, genetically encoded calcium sensor. And it has a lot of the properties of all sensors, which is this, that it's got this calmodulin protein thing stuck on the side of a GFP beta barrel. That exerts some force on that. And what actually happens is this, is that when calcium isn't bound, this sort of opens up the beta barrel and water gets in and quenches, and the fluorescence group, which is this bit in the middle here, I think is protonated, but it goes into a non-fluorescent state. When calcium binds, that thing closes up and this becomes fluorescent. So this is very typical. It's got this bistable state. Calcium bound, it's fluorescent; unbound, it's non-fluorescent. So when you look at your sample, the brightness is telling you the relative amounts of bound and unbound molecules in the sample. And you can then stick that sort of sample on a microscope like this one, which is a spinning disc confocal microscope, so it's a lot faster than point scanners. And here's what you get. So every so often, you will see these coordinated bursts of firing on all these neurons. There we go, there's one. So that's telling us something about the sample. It's not just a bunch of neurons randomly growing. So that's sort of on bioarchive at the moment. The next question is, what is the limit of fluorescence microscopy? What is the limit of detection? So this is a question that Yui Ren and Chris Tate were answering. Do these GPCRs, do they form homodimers, heterodimers? Do they bind? Whatever. Do they know what's going on with these things? That's the question. And they need to see that in live cells because they want to see what is actually happening on this sample. This is what we've got. What we're actually doing, we've got a single protein molecule here on the membrane, and we put a single dye on it, and we've done the same for another GPCR, and for another single one, so we're single molecule, single fluorescent molecule. This is what's known as a sort of bioorthogonal labeling scheme, and this snap tag technology only puts it onto A, and Halo puts it onto B, and Halo doesn't bind onto A, and Snap doesn't bind onto B, and so these are, you can be certain that these things you're labeling two different species. It's live imaging, so we've got to see that against all the autofluorescence from the back of that cell. And one of the tools for doing that is this so-called turf microscope, which only actually looks at things right on the very surface, if that surface is close to a cover glass. That's what the output looks like. And so those blobs are single fluorescent molecules. That's the sort of signal that you can get off that microscope, and those are trackable. So Dina has been working on the tracking of these. That's the limit of sensitivity of fluorescence microscopy. What about I've got a large number of samples to image or a lot of experimental conditions or whatever. We have a couple of instruments that are really well set up for that, a slide scanner, this thing here, or a plate reader, that microscope there. But what they both have effectively are robots to load the sample and software to automate the whole thing. So this one, you put your slides into trays, 6 slides per tray. We've got 10 slides in there. So you could get up to 60 slides on a tray, into the machine. Most of our users are putting multiple tissue slices on a single slide. So you're putting basically hundreds of tissue slices into this thing, you set it all up, press go, and let the machine just chug it through it. So that's one of the real advantages of automation. This one is primarily for plate-based imaging. So we've got a robot that puts the plates onto the microscope, but typically you've got something like cultured cells, 96 well plates, for doing things like screens and high content imaging. So those are very handy things to have. And this is particularly well used in neurobiology. People are doing a lot of work with mouse models. They're getting the brains of mouse models and they're looking for rare things inside this huge tissue volume. By slicing it up and imaging on here, they can find the stuff they're interested in, take those slides and then go off to a different microscope and do higher resolution imaging. Very handy. So this is what a typical tiled image would look like. You can easily get subcellular resolution off these instruments if you wanted, but you get multiple channels of fluorescence and so on. So they're very information rich. So here we have lots and lots of slices load on the trays. You can see they're all identified in image. You can, if you wanted to, you could sort of stack them back up, start to recreate the three-dimensional properties of that brain from it. I'm just going to go on a slightly different tack. This is where Pier was talking extensively about the spectroscopy of flow cytometers. So something like this can go on inside a point scanning confocal microscope because we've got a single point of excitation. The light from there can basically go into a spectrometer, be spread out in the spectrum, and we can do sort of multi-channel imaging off those. And that leads us into this ability to unmix multiple dyes. And so same sort of problem that Pier was discussing. This bit here is a sort of fancy add-on to try and squeeze out some more resolution, but for the time being, I'll just stick to the spectroscopy. So here's something that actually really does happen from time to time. You know, if you were designing an experiment, you wouldn't really want to put EGFP and YFP on top of the same sample, but on occasion, people have transgenic flies or something like that, and they want to look at this, and they want to make a cross between them, and they don't want to rederive them into another dye somewhere out here, but you can sort of see here that it's just not possible to image YFP without a considerable amount of cross-contamination. So in just the same way that Pier was explaining on the microscope, we can basically do detection in multiple bands. So imaging a series of spectral bands. On the Zeiss system, this is all done in parallel. On some of the other microscopes, we have to sort of move the detection bands around a bit in a sort of sequential form. We feed into this machine reference spectra for each of these, and then use an unmixing algorithm to separate the overlapping signals. So we get an EGFP image and a YFP image that is free of crosstalk and bleed through. So that's a property of that was mostly restricted to the point scanning confocals, but recently, PhD student in the lab, Akash, who's just graduated, has taken this into the sort of camera-based system, so an imaging version of this whole thing. And you can sort of see a cartoon of his instrument. So what this is a sort of spectral camera. This device is intended to be plugged onto a microscope where you'd put a camera. So it can go on a wide field microscope, spinning disc, what have you. And it's got all those spectral separation built in with all these filters. And then each of these is a camera, and then you collect all of that stuff. And that's the sort of output you get from this thing. So here if we're imaging here, nuclei, plasma membrane, mitochondria, Golgi outputs them simultaneously. And they're all unmixed. So all of these things are giving massive amounts of crosstalk. So why is this interesting? Well, this is sort of, in some respects, demonstration of what you can do. But we often talk about proteins as their position in a pathway, and proteins that inhibit and cause reactions, turn things on, turn things off. When you do this sort of multi-scale, multi-channel imaging, you have the chance to look at things all together, all at once, because oftentimes the experiments are done in sort of pairwise. What does protein A do when protein B is there? What does B do when C is there? What does A do when C is there? And so on. This allows you to do the whole lot in one go. Right, breaking the resolution limit. So, as I mentioned earlier on, we're basically down at this end. We are limited by diffraction. So there are some tricks around that basically get past that. That's generally called super resolution. So we have a number of microscopes that have this. So in this case, the SIM one, this is structured illumination. We basically use an illumination pattern that can get you a sort of twofold increase in resolution. We go to this one, it basically does the imaging single molecule by single molecule, and that can get you sort of maybe a 10-fold increase in resolution, so down to sort of 50 to 20 nanometers. That's highly dependent on having sufficient labeling density. If you've got sparse labeling, it's not really a resolved image. STED is somewhere between the two. It does that by switching off, by superimposing a donut beam on top of the confocal scanning beam and switching off fluorescence. So the trick with the STED in particular is taking a confocal overview, just draw a little box where you want to do the imaging, and it'll be a lot quicker. This one works with standard fluorescent labels. On these two, you basically have to switch your secondary antibodies. So once you've got a system that's working well, switch the secondary antibodies to dyes that work well either in STED or STORM, where you have to blink on and off, and then you can carry on, do the resolution. I won't do too much more into this, but just to give you an example, there are some systems that do live, the iSIM, instant SIM. This one effectively does the same job as that, but on using a slightly different version of it. And that will actually image live cells at 50 frames a second. This one, this confocal has an add-on that basically breaks down the point spread function into little bits and does a calculation from that to give you extra resolution. There we are. The STED is just that box there. That thing generates a donut beam and superimposes that on top of the regular confocal excitation point. And that's where the sort of magic happens. What do you get out of this confocal? You can get some out of super resolution. You can get something like this. Now you say, okay, I know that's a Golgi, fine. What does the extra resolution get me? And this is a sort of illustration of the point of the power of resolution. Trafficking through the Golgi is all done by vesicles moving through the various leaflets of it in a deterministic manner. This confocal image, you get no sense of that vesicular trafficking whatsoever. If we look in here, in the super res image, suddenly those vesicles are now all visualized, and so you can actually see the really important processes fundamental to Golgi biology, vesicular transport, that just doesn't show up in confocal imaging. Okay, now image analysis, I'm going to do Jerome a great disservice by condensing everything they do into a few slides. But anyway, whatever. You take some images and you've got to try and quantify them. So that inevitably ends up segmenting stuff. Identify all the things of interest and ask questions. What's the expression level of this protein in the nucleus in this cell? So this is where the really important of housekeeping images, you get a reference channel that you can use to segment the feature of interest. So this actually, all this segmentation was done off a DAPI signal and not one of your experimental variables. And please design this in from the get-go because we cannot put those back in once you've got the data. So once you come to ask for help doing the analysis, there's nothing in there that we can segment off that's independent of your experimental variables. Somewhat stuffed. So please do that. I'm going to wrap up on this section here, which is just like everything else, AI is making its impact on light microscopy, particularly in image analysis. And one of the things that is quite well used these days is something called virtual labeling, using a trained neural network. So the general idea is this, is we collect a bright field image, we feed that into a neural network, and that generates a virtual DAPI image in this instance. So what I have to do, I have to train this network. So I have to basically make a real DAPI image sample, a bright field image, use those pairs of samples to do the training. And then when I'm on the microscope, I just collect this bright field image and the network will generate what it thinks the equivalent DAPI would look like. This is very, very handy in live imaging. So one of the big problems we have is the more imaging we do, the more likely we are to kill the sample if we're doing live imaging. Bright field is very gentle on the sample, so you can do that sort of at will. So that gets us past it. DAPI in particular, like every other nuclear binder, eventually is toxic. You've got DNA with molecules stuck all over it. It interferes with cell division, and these cells will eventually die because of it. So you can get around that by doing this. All right, so at this point, fine, we can generate a DAPI image. Well, what actually we're doing in reality is an end-to-end solution. So this is work with Anna and Andrée from the O'Neill group, Ulrika, Jerome, Qimeng, and John Howe. These two, John and Ulrika, previous members of light microscopy group, were working on this use of this virtual labeling method, but taken to its natural limit. So the question of the O'Neill group is, the fluorescent signal interest is delocalized. What they want to do is quantify how that signal is distributed between the nucleus and the cytoplasm during the circadian cycle. So this is 24-hour imaging. In fact, these experiments typically go on for 3 days on the microscope. And these are confluent mobile cells, so they're moving around in time. So the challenges that we have are basically segmenting and tracking both nuclei and cytoplasm independently of this, because this signal level is cycling up and down over time. So we need something else to find the cells. To minimize phototoxicity, we definitely can't use DAPI. That will just kill everything. So doing this bright field virtual labeling technique was the trick. But there is also another thing in mind, which is that if we take a bright field image and slightly defocus the microscope, there's a big contrast change that we can use to tell us about the sample. And so there's a trick which is called phase retrieval, which is just a mathematical operation that you can apply on the difference between these two images to calculate this so-called phase retrieval image. And this is a lot richer in information content. It's got sufficient information in it to allow us to segment out the cytoplasm from the nucleus. So the end-to-end solution is that we can put all of that into a single model. So we're on the microscope, we're collecting the fluorescence channel and the bright field channel, except this time there's two slightly defocused images. All of that goes into a custom trained model, and the model generates the segmentation. So it's not a virtual image. Our output is not a virtual image. Our output is segmentation. And you can see the segmentation superimposed on the bright field image. But what we're interested in is the segmentation on the actual variable of interest, the fluorescence. Now, just to let you know, AI is done. This only does what it's been taught to do. And oh yeah, okay, fine. But actually, I noticed that this video has a glitch in it, and the AI didn't notice. It just carried on as nothing. And so, I mean, only just because I've been staring at this bloody thing making the talk, I noticed it, and it's something to do with the video playback. So I'll have to go back and have a look. But there is a glitch there. AI had no idea. It just carried on. So beware of that. All right, so I think in the interest of time, I'm going to stop and just go to the end. I'll skip the stuff on 2photon. I'd just like to acknowledge all my colleagues who do all the work. Okay, thank you.